So today we have our fantastic speakers. We have Kelly Cumberland. Kelly is an artist and practice-led researcher with extensive undergraduate and postgraduate teaching experience since 2000. It says 200 on the slide there, so that's a very long time that you've been teaching, Kelly. She exhibits nationally and internationally, and Kelly recently submitted her PhD thesis at Leeds School of Design, so congratulations.
Thank you.
We have Heiko Wodak. Heiko received his PhD from ETH Zurich, became an EMBO and AACR postdoctoral fellow in the US before starting his own lab, which is the Stem Cell and Brain Tumor Group, which studies the interface between stem cells and cancer. Heiko is a lecturer at the School of Medicine. And we have Sabrina Samuel. Sabrina received her PhD from the University of Hull and is currently a postgraduate researcher in organ disease modelling. She specialises in modelling brain biology in 3D. So a very interesting group that we have here. And I already have loads of questions. I'm sure you will too. And with that, I'm going to hand over to Kelly.
Thank you. So I am going to share my screen, hopefully. Bear with me. Oh, sorry. Oh yeah, I just need to. Oh. Okay, can you see the first slide?
Yeah, it looks great.
Okay, perfect. So I'll just move to the second. So thank you very much, Dave. It is great to be part of the Horizon series. Really, we're really excited to talk about the research we've been doing and how our exchange has kind of developed over the last 3 years. So in this talk, we are going to demonstrate how generative and cross-disciplinary research offers fresh ways to reimagine biological data. And I'm thinking about Barrard, who discusses this in terms of interaction, the idea that knowledge doesn't come from standing outside and observing, but being part of an entangled process in which materials, people, and ideas shape one another. And this really underpins what we're doing in terms of really being embedded within the laboratory and then the studio and then the exhibition and how we bring in the viewer. So that sense that this goes beyond that kind of art science dialogue that seems quite distant and instead becomes really embedded and entangled through expanded drawing practices, so going again beyond paper into light, silicon and projection. So this space becomes this kind of co-creation opportunity that allows us to develop together and think about ideas differently, bringing different perspectives together.
Yeah, so Sabrina and I are really also excited to be here. And we think it's kind of a prime example of not interdisciplinary, but cross-disciplinary work where the science as well as the art stays true to their methods. And we basically view the different field and contributed to it with our expertise. And as Kelly said, challenge concepts, trying to find fresh perspectives. And in order to do this as a starting point also for this talk, and what we like to do as scientists is we like to find a baseline, so-called negative control. And our negative control here is what we absolutely do not want to be. And we want to be in a very different space to science illustration. Because what's happening here is that, familiar cliches are used to already create an image that is expected in a human brain. When you read the title of a, let's say, slightly sensational science news article, what if structures that we grow in the laboratory that mimic the biology of the brain, what if these at some point become conscious and feel pain? And of course, this, together this works very well and it has its merit. But like imagery like this, I would say like I'm not the only scientist that says like it's a bit alienating because there's really nothing truthful in this image. And what we are trying to do in our collaboration is almost find an integrity between the two disciplines. And what we in the lab are looking at is something like this. So we use microscopy, and we can zoom into different scales. So we can look at biological building blocks. On the left-hand side, this gray image is electron microscopy. So zooming really, really deep into the contents of a single cell. We can isolate materials, get protein structures. There's work in, Kelly, if you can go back one slide, please. There's work that Kelly will show, like looking into inspired by protein structures, but we can also look at the mesoscale cells interacting with each other. And then from there, we can try to build some really complex models that are sometimes called miniature brains, what you see on the right-hand side. And you can trust us, we have profiled those extensively. They don't have pain receptors and they don't have consciousness. And what we have to do is, scale is treacherous in biology. So the biggest object on this slide is actually the mini brain in the corner, which isn't obvious when you look at things, and art can ignore this scale. We always have to measure, and we have to isolate events to get to a quantitative outcome.
So here's an example of isolating these events. I'm really zooming in on cell behavior. We won't be able to see this in sort of a holistic view. This is something we have to zoom in, isolate the cells and look at these behaviors individually. Yeah, like Heiko said, in order to get a quantitative output. But this could be quite isolating. Even in the room doing this, you sometimes forget sort of like the materials and reagents that you're working with. We sort of think of them as quite mundane because we use them every day. Even the materials themselves could be quite interesting. So such as saline solution that we discovered was quite interesting when Kelly came for one of her first visits.
Yes, so let's begin with this early material experiment. So I was using saline solution. And this is things, as Sabrina said, that are kind of background noise for the scientists. It's just part of a daily routine, something that's just used. And as we were spraying the laboratory down before an experiment, I started to think about the saline solution as an intentional process as opposed to that, well, unintentional, I suppose, bringing those two ideas together in terms of a generative, playful engagement with the materials that are used in the laboratory to clean it. So the saline solution evaporates and crystallizes, and these traces start to form. And this made me also think about vibrant matter in terms of Bennett's idea that helps to explain that sense of notion that materials have their own vitality. And then this kind of the salt, the air, the surface, how they all participate in the making. So this is really about how this becomes observing through transformation and depiction, and not about how we represent something directly. So let's clarify three key terms to anchor this discussion. Negation refers to the absence of and fragmentation, the gaps, erasures, or accidental marks that disrupt a drawing's intended narrative and create tension and meaning.
Another term we'll talk about during the session is co-emergence and co-creativity. So this describes, in this situation, the art and science groups working synergistically and creatively to create something greater that would have been created in isolation.
And this brings us to the really important factor of serendipity and I think also curiosity. I mean, science history is basically full of little accidents, like wrong or supposedly wrong buffer compositions or wrong microscopy filters that led to really valid discoveries. And And also to mention that in science, mainstream science philosophy, as per Karl Popper, we're trying to prove our hypothesis wrong, which brings us back to negation.
So when I think about negation in my drawings, I don't see it as a loss, but a productive absence. So this is a space where something new can take shape. And Griffiths calls this a sideways look at time, which feels close to how expanded drawing works in the sense that it's continually folding back into itself, looping and reforming. And this kind of unfolding through difference brings in ideas of Derrida in terms of the work is never complete. And in a way, the audience completes the work by the way they engage and walk around it and view it from different angles and start to find different drawings within the work itself. So it has this sense of unfinishedness. And that sense of experimentation and the idea of drawing with, which also expands the sense of the use of materials themselves, as well as the subject and as well as the processes, how these ideas relate to Ingold's sense of drawing with as opposed to drawing of.
Yeah, so we use sort of negation, like I said, in a scientific context as well, pretty much all of the time. So 90%, 95% of experiments don't create positive results. A lot of the time it is sort of trial and error and learning things from negative results. And this is, I think, a culture in science which is sort of frowned upon sometimes. Because obviously when you try and publicize data in the lab, you're encouraged to create a narrative, like a story that has an exciting positive result at the end of it. And so a lot of the time, all of these negative results, which is predominantly what's been created, are just ignored and can't necessarily be used. Maybe you get a negative result in the lab, but because it brings about an area of biology that you're not really particular with, you don't look into it anymore. But I think there's been a slight change recently to try and publicise more of these negative results so we can learn and maybe spark new conversations and new lines of inquiry from these sort of like negative spaces. So an example of that is there's a journal that's been brought out in 2020 that's been becoming more and more popular recently called the Journal of Trial and Error. And this is where they try and encourage this culture of publicising negative results so we can actually learn from these spaces. So here's an example, a visual example of what's some results we can get in a lab. So give a bit of background. So in this process, we've taken some pieces of tissue, or in this example, it was an organoid, so one of our mini brains. We've fixed it, so we've held everything in position, we've sliced it up into thin slices, and then we've gone ahead with antibodies that will bind to proteins that we're interested in. And then these antibodies produce light, and then we can extract this light using a microscope. And then we change all the colours to make it visualise different interactions. But yeah, this is an example of how our data can be visualised, although it's semi-artificial. So these colours, those proteins, they aren't green. That's just a happy accident of the process we used through the methodology to get these images.
And once we have these markers, we can then quantify the cellular behavior. And for this, we use the principle of image segmentation. And there's free software for this. For example, here a tool called ImageJ, which we frequently use, where you can basically, again, and here the theme again is emerging, zooming in and isolating events against the artifacts that this segmentation or the segmentation algorithms can produce. So we have to be, again, very, very selective to say, like, okay, do we have the right cells in our quantification? And we focus in more and more to get our result and to get our comparisons. Now, image segmentation can be used in a very different way artistically, and this is then very exciting for us to see when artifacts actually don't matter and we don't need to focus on a quantification.
So using the images of segmentation, I was continuously redrawing, thinking about the isolated data, and this created new discussions and developed our shared language, actually. This was a eureka moment in the laboratory where I brought these images in, and we had this moment of bringing these 4 aspects together and starting to think about them from a different perspective in terms of the drawings I'd made, and how they start to, instead of mirroring back, but instead they kind of bend through difference. That it's about seeing how light, material and perspective interfere and create new patterns. So really thinking about the observational spaces and how this emerged through this entangled perspective. So in this sense, each time I was reworking, reinterpreting, reprojecting, using different data, using different types of absorbance in relation to a machine in the laboratory and how it records absorbance of cells. So this starts to kind of translate the ideas through the laboratory into the studio and back again. And this sense that art and science can diffract through each other, generating this layered kind of development of language and knowledge, rather than perhaps presenting a single answer. And Barrod's idea of entanglement fits this. Our observations are shaped by how and what we choose to look at. And these kind of redrawings feed that process back to us. So we're really engaged in this feedback spiral as opposed to a feedback loop. We're constantly moving forward in the same way that an experiment would develop and not happen twice in exactly the same way. So there's this kind of spiral feedback that has formed through our discussions. And this kind of really allows the collaboration to develop in a ongoing way, as we constantly find new things and discuss how we might see things that potentially are overlooked within the laboratory, and from an artist's perspective, how I see them differently, and vice versa. So just to think about the techniques that we're talking about, in a sense, there's iterative layering. So this is offering reinterpretation of data and chance. So again, bringing that sense of chance, what the layering does, how it interrupts the image, and responding to this directly through our discussions, which again always feeds back into the work. Projection then expands this. So scale is very important in terms of observation, and how scale absolutely changes what we see in the laboratory into an exhibition space. By projecting into this kind of opportunity, gives us a spatial logic that brings a different thought or thinking or engagement even, and especially in terms of how we view and discuss the work. And fragmentation also invites participation. So that idea of separation, isolation, dissecting, and how these kind of layers start to build a dialogue through art and science. So this is, transformation of processes rather than a depiction of ideas.
Or do I go back one? I think we skipped one.
Oh, I've skipped one. Sorry.
All right.
Yeah, sorry, Heiko.
Yeah, so I really like this one. It's not moving at the moment, but so we set out
Is
from
it
the
not?
beginning. No, it's not, on my screen. It isn't. You have to play the movie, yeah. So we set out from the beginning. We said, like, we don't want to create necessarily anything beautiful. You know, like it's really finding finding an integrity in the disciplines. But sometimes something beautiful comes out. At least it's always a matter of taste. But I look at this and so many things come to mind. Potentially, so this is like here are our technical and biological replicates. And as Kelly said, like iterations, no experiment that we do in the lab is ever the same. The cells will always be in flux. There's always a change, and there are always, and we had it before, like error bars. And to me, all of this is, like a feedback loop that comes from this. I know where it came from, right? The cells under the microscope. It's like so in a way abstract, but also like, reminding us of this flux that we often disregard when we look at static images and focus in on our quantification. That's fantastic.
So here's an example of Kelly using a set of an image, a single image from the lab. So these were endothelial cells that I grew in the lab, and actually I thought, so this sort of dark section in some of the images you can see was actually a little bit of condensation from the flask. So I would usually sort of disregard this as like an error or something that's just in the way of my quantifications. But it's actually become quite a significant sort of centerpiece of this piece of art. And it's quite an example of something we'd normally disregard could actually bring about something quite important. So when I sort of look at this, I think of the condensation being on the top of the flask could bring about thoughts that could start a new experiment. So you think about the condensation on the flask, that's become about because of changes in temperature within the media. So why has the changes of temperature come about? That could be because of changes in metabolic activity within the cells. And this slightly abstract, but could spark a new experiment, and something that I wouldn't have necessarily thought about if Kelly hadn't come in and pointed out how these could be a focal point.
And then fragmentation extended this. So this was another point Sabrina and I were looking at the screen, and there was this kind of computer lag. So these images were starting to appear. So the top one's the computer image, and then the other two are my kind of translations and explorations. So observation here becomes temporal. So that sense that that kind of completes itself, this movement and change of an image then offers a new image and a new way of thinking through something. And also, again, something that I was pointing out as quite an exciting moment, and something that was just part of it just happens because there's a lag kind of situation. So we had a conversation around those kind of, again, things that are not, that are kind of peripheral to the focus of the experiment. And this kind of really brings that idea of becoming and articulates this, thinking about Deleuze and Guattari's idea of becoming, this kind of the fact that knowledge isn't an endpoint, but a process, and it's a continual transformation. So each iteration, whether a visual experiment, whether visual or experimental, does alter how we think, and it alters how we then think about the next experiment. So I'm just going to quickly play a video from Light Night, which has a bit of sound. So I'll leave that on, and then we will start to have some final thoughts. Is that very quiet?
Yeah, we can't hear it.
Oh.
I don't think. Oh, actually. So I think maybe you might need to unshare and then reshare with sound.
Okay. Shall I do that now, or we could carry on talking, and then we can,
Okay.
I can bring it up while we're in the questions if that's easier.
Okay.
Okay,
I think Light Night was really what brought it to life was the audience. I think for the basically first time, the like we are not just two observers of objects where we sit in rooms and after lab meetings, but it's in a real exhibition space and people are walking through. And they also don't necessarily, people at Light Night, of course, come from many different exhibitions, so they don't necessarily expect an art science exhibition. And I think this is really then a space that changed things, how also we ourselves interacted when an audience was there, right, Kelly?
Yes, yeah. And again, as I have said, this does bring in this kind of third observer along with ourselves. So the audience really interacted with it in very different ways. And there was this sense of kind of looking behind things, which is, I wanted people to really kind of physically engage and move and look and bend down and crouch and find the work, because it wasn't all necessarily visible on first sight. You had to kind of move in order to explore it and use your body as a way to do that, to in a way finish the work itself. So this kind of opened up those ideas that we're talking about, observation and ways of looking, ways of seeing, ways of thinking, and finding something that perhaps you wouldn't have necessarily found if you just sat in one position or looked from one perspective. And this particularly thinking about the assembloid here. So again, movement, scale, change, reflection, how these ideas were then translated through this drawing. And interestingly, the hole in the floor, or the mirror actually, not a hole in the floor, got a lot of attention, especially children. They were constantly stamping on it, trying to figure out what was happening there and if it was a hole. So this kind of allowed the work in a way to become alive with the use of light and movement and change, and also that sense of people, as I say, having to look in order to see, but look in different ways. So using expanded drawing, perhaps as a model of a way of thinking through co-emergence, where different forms of knowing grow together and embrace uncertainty and open-endedness rather than completeness. So kind of offering this point of engagement for the viewers as well, where everything's kind of in flux. It's also about trust, actually, the sense of giving space for uncertainty, for people to kind of engage with these provisional moments and kind of questioning the ideas and becoming part of the discussion.
I guess so the reason why that piece was spinning was it was trying to sort of recapitulate what we see in the lab. So here is one of our mini brains that we developed, but interacting with some brain tumor cells that we fluorescently labeled so that we can sort of track their movements and observe this interaction. But that actually, the movement that you see in here is something during the quantifications of this, so we can compare these interactions in different conditions. We sort of removed this movement and we subtracted it during the algorithm. And I think it was quite sort of special to see how art was able to sort of put back these missing parts that we took away through science.
So this kind of sense of how art can kind of bring ideas together that science perhaps is, because it's focused very much on the experiment, the outcome, and bringing those kind of moments of the process, the unseen, those eureka moments within parts of the process, not necessarily the final moment. So this kind of rotation has continued as well through this work, and the two drawing, well, the drawing of mine on the top left and the laboratory image bottom left. So these works were developed at exactly the same time. And independently we shared them at a laboratory meeting, which raised conversations about this kind of intuitive exchange within our research and how working together over 3 years has started to kind of develop this visual language and moments of intuition and change. And yeah, it was quite a kind of moment of eureka, what's going on here? So yeah, we're kind of exploring this at the moment. And I'd like to just invite you to an upcoming exhibition, which is Assembloids Drawn Alive. So the work will invite viewers to look again and to really engage with the body of work that's been culminating over the last 3 years, and to really kind of think creatively about biology and intertwining acts of continuous becoming. So there will also the exhibition is open 24th of November to 23rd of December. That's my error, sorry. And there is an exhibition event on the 4th of December, 4 till 7. So I will be sharing that on socials. And we're also going to be doing an in-person talk in the exhibition, the 3 of us, so we will publicise that as well. And just to quickly add, while I'm giving a quick plug, we also have an exhibition at the Atrium Gallery, Bexley Wing, February 2026 as well. So please do look out for that. I'll share information. So there are contact details, and that's the reference list. Thank you.